So, in the nucleus of eukaryotes, chromosomal DNA is packaged into nucleosomes, which are essentially repeating units of DNA wound around conserved proteins called histones. Okay. Two of the histones that mammals produce are H3A and H3B, which differ only slightly in amino acid sequence and are encoded by separate unlinked genes. H3A and H3B function during particular points in the cell cycle. Okay, so right now we know that those two proteins are important for the cell cycle. Scientists hypothesize that histones H3A and H3B play roles in the growth and development of diploid cells, cells that divide to produce gametes. So they're important for development and the growth of diploid cells. In order to investigate the relationship between the functions of H3A and H3B, scientists created two groups of mice. One group was heterozygous for deletion in the gene encoding H3A, which will be called H3A plus minus. And a second group was heterozygous for deletion in the gene encoding H3B, which is called H3B plus minus. Plus means present, minus means absent for the allele. So plus means functional allele, minus means not functional allele. So far so good? Okay. So let's go ahead right now and do A. Describe the expected genotype ratio of offspring from a cross between two mice that are heterozygous for a particular gene. Okay. So they're heterozygous, right? That means they have one positive copy and one negative copy. It's your choice to mate this one with itself or to mate this one with itself. It won't affect anything because they just want the genotype ratio, basically. Okay? So look, right now if I take H3B plus minus with H3B plus minus. Right now, let's go ahead and get the gametes. This will be, one gamete is going to be H3B plus, one gamete is going to be H3B minus. One gamete, H3B plus, one gamete is going to be H3B minus. Right now, if I do the Punnett square, what do I expect to get? So do the Punnett square. H3B plus, H3B minus, H3B plus, H3B minus. Shall we do it? So H3B plus minus, H3B minus minus. H3B plus plus, H3B minus plus. So what's going to be our genotype ratio? We're going to have 1 plus plus, 1 minus minus, and 2 that are plus minus, right? So we're going to say 1, 2, 1, okay? And for 1 H3B plus plus, 2 H3B plus minus, and 1 H3B minus minus. That's going to be our genotypic ratio, essentially. Okay. Good. Now, the scientists performed a cross between H3 AA, okay, mice, and that are also H3B plus minus, and determined the H3B genotype of the offspring. The scientists performed another cross between H3B plus plus mice that were also H3A plus plus, and determined the H3A genotype of the offspring. They then determined the mass of each of the offspring. Mice that were heterozygous for deletion, that means having H3B minus minus, died during development. Okay, so what do we see on the y-axis? Average mass. So average mass of the mice was essentially our dependent variable. Okay, you see, our wild type is going to be the control. Why? It has no mutation. Okay, and if they were H3b minus minus, they died. That's why here H3b minus minus has essentially no mass because they just died. Okay. They were doing this on 3-week-old mice. Okay. BI, identify the dependent variable in the first experiment shown in figure 1. Figure 1 is here. So what's going to be dependent variable? The average mass of the mice. So the average mass of the mice. Okay. Double I. Justify why the scientists used the mass of the wild type plus mice in the first experiment as a control. So as you can see, they did use plus in both genotypes. So they had plus for the H3B genotype and also plus for the H3A genotype. Well, the control is always going to be good, right? Because how do you know if this mass is low or high? How would you know if it's low or high if you have nothing to compare it to? Okay, so we're going to say that it provides a baseline to compare the mass of mice to from genotypes that were plus minus or minus, okay, to see the effect of single/double deletion on the mass of the mice. Okay. Okay, triple I. Justify why the scientists used mice of the same age for the first experiment as another control. Well, if you use mice of the same age, this allows you to eliminate age as a possible variable that might interfere with the interpretation of the results, right? So to eliminate age as a variable that may affect the interpretation of the results. Okay, so to make sure that mice mass differences are due to genotype deletions only. Okay. IV, based on data in figure 1, describe the effect of losing one functional H3A allele, so having H3A plus minus, compared with the effect of losing one functional H3B, which means H3B plus minus. So look, of course, you must always compare to the control. You must compare to the double plus. So go back here and look what's happening. So if the mouse has a plus minus for the G3P, how is its mass compared to the control? Less or the same or more? It's less. So we can say that the H3B plus minus genotype led to less mice mass compared to H3B plus plus. Can we say the same for the comparison of the plus minus of the H3A to the control? Are they different or the same?
Different.
They're the same because the error bars are overlapping. But the H3A plus minus genotype has no difference in mice mass from the H3A plus plus. Okay. Good. The scientists then examined the structure of the gametes made by each of the offspring to assess the relationship between genotype and meiosis. Data for the analysis of sperm made by the H3A double plus wild type. So the wild type will always serve as the control. And the H3A minus minus mice. Who here is better? Looking at the data. Who here has less defect?
The plus plus.
Yes. The plus plus, the majority of the sperm has no defect. But the majority of the sperm in the case of H3A minus minus has tail defects or head defects. Based on figure 2, describe an effect on the sperm structure of the H3 minus minus genotype. How would we describe what's happening here? It has more defects in terms of tail defects, mostly, and head defects.
That's the answer.
Yeah, that is. CII, assuming the scientists examined 200 H3 minus minus sperm, calculate the approximate number of H3A minus minus sperm that had structural defects. Okay, so tail defect and head defect both count as structural defects.
Yeah.
How much will these be?
Add them up.
Yeah. So first of all, how much of the sperm had no defect?
About like
15% was without any defect. But the 85% left had a defect. So now what do we say? 85% had defect, 85% of 200, which is 170 sperm. So out of 200 sperm, 170 have structural defects when the genotype is going to be H3A minus minus. DI, state the null hypothesis for the effect of the genotype in the second experiment, data shown in figure 2. The second experiment is about the sperm structure comparing H3AA mice and H3A minus minus of the same age. Again, to eliminate age as a possible factor that might affect your interpretation of the results. So, null hypothesis is always about trying to unlink two things, right? So, null hypothesis is going to be that deletion in the H3A genotype has no effect on defect percentage in sperm. Yeah.
Do all these answers look the same? Because
Yeah.
all of them I've done goes against it.
Yeah, for null hypothesis, it's always the same. For control, it's always going to be the same, and so on. DII. Scientists claim that having two wild-type H3A alleles cannot restore the function caused by deletion in the H3B. Okay. Using data from Figure 1 support the hypothesis. Two wild-type H3A. H3A plus plus cannot restore the function caused by deletion in the H3B. So H3B minus minus. Let's see. Look for H3A plus plus and H3B minus minus. Even though there is H3A plus plus, it could not help when the H3B is minus minus because they all died, right? So we're going to say that even when the mice were H3A plus plus, having H3B minus minus led to zero average mass due to death of the mouse. Good. Human ribonucleotide reductase HRR enzyme helps maintain the supply of available nucleotides in a cell, which is essential for DNA replication. A. Describe the three structural components of a DNA nucleotide. What do you guys think?
The phosphate, 5-carbon sugar, the nitrogen bases.
The phosphate group, the 5-carbon deoxy ribose sugar, and 4 different nitrogenous bases. So A, T, C, G. Now, some commonly used anti-cancer drugs, while effective against cancer cells, often cause damage to healthy cells. In a search for cancer treatments that cause less damage to healthy cells, researchers identified the small molecule NSAH that can reversibly bind to the active site of HRR and inhibit the activity. So essentially, NSAH can bind to HRR and can inhibit its activity. I use the symbol to mean inhibit or to block. To determine the most effective concentration of NSAH, researchers measured the activity of HRR in the presence of 6 different concentrations. Okay, what do we see in here? With more concentration of NSAH, the amount of HRR is dropping significantly, right? So more NSAH means less HRR. BI. Using template in the space provided for your response, construct a line graph to represent data in table 1. Your graph must be scaled and labeled. So, before we do the graph, it's important to take care of the error bars, meaning the plus and the minus. So let's go ahead. 98, 102. 90, 106. 86, 98, 47, 63, 33, 4. Do you guys know what I'm doing right now?
Mister, I don't get it.
Look, you see how it's plus minus 2? So I do 100 minus 2, 100 plus 2. And they're same for the rest. 20, 28. Now it's time to graph. Okay, so what goes on the y-axis? The dependent variable, which is the percent of maximum H R R activity. What goes on the x-axis? The concentration of NSAH. Write the unit, mu molar. Now, the concentration ranges from 3 to 100. So if I can do this as 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, I have now successfully covered 2/3 of the x-axis, which is perfect. How about the y-axis? My lowest number is a 20, and my highest number is 106. If I do here 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 110, I should be fine. Okay? Now, so 10 all the way to 110. And this one is going to be, we said 10, right, as well?
10 to 100.
Yeah. So, when it's 3 for the concentration, it's 100 for the activity. So 3 is here, or here. 100 for the activity, which is here. So I'm going to do this. How about the error bars?
It's gonna be a line graph.
Yeah, we were told to do that. 90 to 102. That's 100. That's 90. So 98 to 102. And connect them. That's my first point. My second point is a 5, which is here, and a 98. So a 5 and a 98, which is going to be here. Okay. And we should go from 90 to 106. So 90 to 106. Okay. Next up, 10 with 92. So 10 with 92, which is going to be here. Error bar ranging from 86 to 98. 86 to 98. Next, 25 with 55. So 25, and this is 60, 50, 25 with 55. Ranging from 47 to 63. Then 50 with 41. 50 with 41. Ranging from 33 to 49. So 33 to 49. Next, 100 with 24. So 20 is here. 100 with 24. Ranging from 20 to 28. Line and graph, that means you must connect them all by lines. 1, 2, 3, 4, and 5. And that's going to be what my graph should look like. Okay. Yeah, I don't need this anymore. Okay. Now, double I, based on data in table 1, determine the measured concentration mu molar of NSAH that resulted in a greater inhibition of HRR activity than was observed with the 25 mu molar NSAH. The 25 molar ranged from 47 to 63, right? So which concentration resulted in more reduction, which means less than that, with no overlap? 100, right? Because if you choose 50, you would have overlap in that with that. So what do? The 100 has no overlap. Did you guys get that part? The overlap, do you know? I can explain again. So look, this one here ranged from 47 to 63. They're asking for more inhibition, which means less HRR. This one will range from 33 to 49, okay, which is going to be overlapping with 47 to 63 because they both have 49 in common, or 48 or 47. So don't pick this one. This one's going to range from 20 to 28, so no overlap.
You can also just look at the graph.
Or the graph, yes. So on the graph, as we can see here, this is the 25. That's overlapping with it. So don't choose it. Okay, you must choose one that is not overlapping with it, with less concentration of HRR. Less concentration. It has to be lower than it. And this one here is not overlapping with that one. Okay, so the 100. Okay. Based on data in table 1, identify the two measured concentrations of NSAH between which the HRR activity was 75% of the maximum. So look, the maximum HRR activity was 100%. What you want to do is to choose 75% of that. So two concentrations of NSAH between which the HRR activity was 75%. Okay? So what are those concentrations? Can we tell?
10 and 25?
Yeah. So between 92 and 55, there has to be a 75, right? So between 10 and 25. So between 10 mu molar and 25 mu molar of NSAH. Okay. DI. Among individuals with cancer, some individuals produce twice as much substrate for HRR as others do. Predict the effect of this increased substrate concentration on the amount of NSAH required to be effective. NSAH is supposed to be blocking HRR. That means if you produce more HRR, you must also require more NSAH to block it, correct? So we're going to say more concentration of NSAH is needed. Okay. Now they're saying justify your prediction. Twice as much substrate for HRR as others do. HRR was the enzyme, correct? And now you're going to have substrate that is competing with NSAH. They both compete to bind to the active site of the HRR. So if you have more substrate, you must have more NSAH to outcompete this amount of substrate. Okay? So if you guys look back at the question, we were told that a molecule, NSAH, reversibly binds. That means it's called a competitive inhibitor. It has to compete with the substrate already there. So we're going to say that NSAH is a competitive inhibitor of HRR. So it must compete with the substrate binding to its active site. Okay. To actually bind. So more substrate means more NSAH needed to compete.
Did you write it down?
More NSAH needed to compete. Okay, F2C, a commonly used anti-cancer drug, functions as an irreversible inhibitor of HRR. So it's going to bind. So unlike NSAH, it's irreversible. That means it does not have to go through competition with any substrate, which means it's going to be more effective. Explain why treatment with a reversible inhibitor like NSAH might cause less damage to healthy cells compared to a reversible one like F2C. So F2C binds to allosteric site of HRR and doesn't need to compete with a substrate. So even at low concentrations, F2C can block HRR's activity. While NSAH must always compete with substrate to bind to HRR's active site. Okay. After being infected by a certain virus, host cells produce a viral protein called SEDV, which then becomes embedded in the plasma membrane of the host cell. To become fully activated, SEDV must undergo. I'm having a hard time reading.
Endocytosis.
Yeah, my eyes are like. Must undergo endocytosis from the plasma membrane. So it has to enter into the cell. After endocytosis, SEDV is cleaved. That means it's going to be broken up into two fragments by a host cell enzyme. These fragments are then embedded in the plasma membrane, where they will become part of future virus particles, and they can also facilitate future infections. You know what's happening here? The host cell is trying to defend itself from the virus. It's going to cleave the viral protein, but the protein of the virus actually wants this cleavage. It wants that. Because now it can attach to plasma membrane and can facilitate the future infections. So the cell inadvertently, that means unintentionally, is helping the virus. SEDV contains a sequence in which many of the amino acids are tyrosine. To determine whether this tyrosine containing sequence is required for SEDV to undergo endocytosis, once SEDV is embedded in the plasma membrane, researchers created several different SEDV mutant proteins. So now we have several different SEDV mutant proteins, where the amino acid alanine was substituted for increasing number of tyrosine molecules. The researchers then used green fluorescent tags to label SETV proteins that remained embedded in the membrane, and red fluorescent tags to label SETV proteins that were taken in the cell. So red means in the cell, green means on the membrane. The researchers compared the ability of cells to take in the different mutant SETV proteins by endocytosis with the ability of the cell to take in the wild type. Right, this wild type is going to serve as our control. A. Describe the structure of the eukaryotic plasma membrane. So what is it made of? The plasma membrane. Do you guys know?
Yeah.
Can you enlighten us?
It's either proteins or phospholipids. Something like that.
Let's just stick with the phospholipids.
Okay.
2 layers of phospholipids with hydrophilic,
Hydrophobic
heads
regions.
facing outward and hydrophobic tails facing inward. Okay. Okay. B. Justify why the researchers compared endocytosis of the mutant SETV proteins with endocytosis of the wild type SETV protein as an experimental control. You must have a baseline. You must, right? Right now you go and you take a sleep medication and you sleep for 8 hours. Is that a lot or not? You must have people who sleep without the medication and see how much they're sleeping to compare yourself to them, right? The control is the same thing. So this allows the researchers to attribute any differences from the control to said V mutant proteins. Okay. C. Researchers hypothesize that as the number of alanine substitution in the SETV protein increases, the likelihood that SETV will undergo endocytosis decreases. Based on this hypothesis, predict the relative amount of the green and red fluorescent the researchers will observe as number of the alanine substitutions in SETV protein will increase. Guys.
So green will be more.
Yeah, because green means what? They're going to stay in the plasma membrane. So guys, green means what? Plasma membrane. Green plasma membrane. Red inside the cell. Okay, but now you're going to have less endocytosis. That means more will remain in the plasma membrane. So you're going to have more green than red fluorescence. Or more green and less red. Okay, D. Researchers claim that the portion of the SETV protein that becomes embedded in the plasma membrane is composed primarily of amino acids with hydrophobic R groups. Provide reasoning to support the researchers' claim. The interior of the plasma membrane is a hydrophilic or hydrophobic? Made of what? Made of fatty acid tails. So we can say that the interior of the plasma membrane is hydrophobic, made of nonpolar fatty acid tails. So the nonpolar or the hydrophobic R group can interact with these tails. White bark pine trees are native to high elevation mountain ecosystems in North America. The seeds of the white bark pine tree are contained in pine cones that are covered with thick scales, which are essentially hard outer layers that protect the seeds from the harsh conditions of these high elevations. In order for the seeds to be released, a small bird known as the Clark's nutcracker must break apart the scales. The nutcrackers eat some of the seeds immediately and bury others in different locations, where the nutcrackers may eat them at a later time. In recent years, the number of white bark pine trees within a population has decreased dramatically due to several factors, including an infection caused by a non-native fungus and also an infestation, which means the presence of an unusually large number of mountain pine beetles, both of which will cause the trees to die. Guys, just logically, if you're killing the trees, they're going to have less seeds. And the birds that eat the seeds will now starve to death. Would you guys agree?
Maybe not starve to death, but decrease in
Yeah, not starve to death. Yeah, I'm sure they can. But I'm sure you guys agree. A. Describe the relationship between the density of the population and the effect that competition has on the growth rate of the population. So, if density of the population increases, will it suffer from less or more competition?
increases.
Huh? More or less, choose. You gotta choose.
What did you say?
If we have more density population, do we have less or more competition?
It's more. So
Yes.
less.
More so less?
More.
More. Yes, more gives you more. Look, if you have more crowdedness, you're gonna have more disease, more competition,
okay.
and everything else.
That's easier to say.
More population density leads to more impact by density dependent factors. So more competition for food, space, and mates. So less growth rate of that population. Okay. B. Explain how the nutcracker's behavior of releasing the seeds from the pine cones affects the number of the white bark pine trees within this population. So, spreading of the seeds outside of their original area, is that a good thing or a bad thing?
A bad thing.
Why is that?
Because
It's actually a good thing.
Why? It's not their environment. It's not high elevation. How will they survive?
Because maybe there's already too many of them in that same area. But now, if you bring that new seed to a different area with less competition, it's better for them. You know, it's always better if you spread the seeds outside of their environment. It's always better. Okay. It's like if if all your neighbors are your family, is that a good thing?
No.
You're gonna stay in the same mindset. You must leave that mindset. You know what I mean? Okay. So basically, it's better for their numbers.
You can just write that in the exam.
Well, I didn't finish yet. as the seeds may land in new areas with little density of the white bark pine tree, so they face less competition and more chance to survive. Okay. C. Predict the effect of a continued increase in the number of mountain pine beetles or the number of the nutcrackers within the population. Those pine beetles, were they a good thing or a bad thing?
Bad.
They were eating the plant, right? So they lead to decrease in the number of nut crackers. Okay, so they're gonna say justify your answer. We're gonna say that the mountain pine trees eat the white bark pine trees. So this means less seeds for the nutcracker birds to eat. So pitcher plants are plants that digest insects to obtain nutrients necessary for production and growth. Scientists studying the diverse morphological features of pitcher plants created a cladogram representing an outer group. Do you guys know what that is? Is the least related group to the rest. It has the least things in common with the rest. Okay? And the three clades of pitcher plants, A, B, and C. The cladogram includes the evolution of 5 traits: digestive glands, wax crystals, lids, plants have lids, transparent spots, and also slippery surfaces. Very interesting. A, describe one way a mutant allele can increase in the frequency in a population over generations. Any input? If the allele increases the survival of individuals, they will survive and reproduce and pass on this allele under the influence of positive natural, because look, there's positive selection, there's negative selection. Positive selection means you're going to be increasing it. Okay. B, explain how the cladogram in figure 1 shows evidence of convergent evolution of pitcher plants in the clades B and C. So clades B and C, right? They both have, what do they have as
closest to each other?
Okay. So look, they both have slippery surfaces and they both have transparent spots, correct?
Mm-hmm.
But both of these traits appeared within the clade. So not before the nod, but after the nod. And they also appeared separately, or essentially independently. This means that these traits appeared not because of the presence of a common ancestor, but because of the presence of similar environmental pressures that led to the evolution of the same feature. Okay, so we're going to say that traits 4 and 5 appeared past the node. This is called node, from which clades B and C appeared. So they are not due to common ancestry, but they appeared independently due to facing similar selective pressures. Okay. C. Wax crystals are ancestral to clades A, B, and C. But they're no longer present in any of the species of clades B. If you guys look at the clade B, there is no wax crystal. They appeared here. But they are no longer there in a clade B. So that means they were taken out at some point. Represent the point at which the wax crystals were lost by adding an X to the cladogram. So you should put the X here on clade B, because clade B lost them. X means they lost those wax crystals. Okay. D. The lids of pitcher plants in clades A, B, and C have different morphological characteristics. Using figure 1, explain how divergent evolution could be responsible for these differences. So, what is the meaning of divergent evolution? They would have different morphology despite having a common ancestor due to different selective pressures. It's actually quite the opposite of convergent evolution. Okay? So we're going to say that they evolved or they originated from the same common ancestor, but due to different selective pressures, they basically evolved different morphological traits. Okay. And let's look A, B, C. So A has 4, B has 4, and C has 4, right? But A doesn't have 5. Would you guys agree? Yeah. So lids of pitcher plants have different. So the lids, so they're actually no, that's it. That's perfect. Because if I say their lids have different morphological characteristics, I won't get extra points. Never repeat what they say. They don't like that. And if you do, you just waste your time. Small RNA molecules can regulate transcription and translation. Scientists investigated the relationship between a small RNA molecule known as miRNA 466 and the expression of a gene that encodes the growth factor GF. The expression of the GF varies in response to oxygen levels. Scientists observed that deleting the DNA that encodes miRNA 466 from the genome of the mice results in less levels of GF protein. So basically, if you delete the DNA that encodes for miRNA 466, that means miRNA 466 will decrease, which means that GF will decrease as well when there is low O2 conditions. To determine which stage of gene expression is regulated by miRNA 466, scientists harvested cells from the mice lacking the DNA encoding miRNA 466. They exposed the harvested cells and control cells to low oxygen conditions for 24 hours. Then they measured the amount of GF pre-mRNA and also the GF mRNA produced by the cells under both normal and low oxygen conditions. Okay. A. Based on figure 1, describe the effect that low oxygen conditions had on the production of GF pre-mRNA in cells without miRNA 466. So go back to cells lacking, so these guys, under low oxygen conditions. The amount of the GF pre-mRNA. Is it less than the control, or is it more, or is it the same? How? How do you see overlap between this?
Oh, this. I thought you were talking about another different. It's different.
It's less, right?
Yeah.
So they produced statistically less amount of GF pre-mRNA compared to the control. Okay. B. Based on figure 2, describe the effect that deleting the DNA encoding miRNA 466 had on the production of GF mRNA in low oxygen conditions. So again, go to low oxygen conditions and go to this one. Compared to the control, did they have less or more amount of GF mRNA?
Which one? Less.
In the low oxygen conditions, they also had less, right? They had significantly less amount of GF mRNA compared to the control. Okay. C. Scientists claim that miRNA 466 promotes the transcription of GF in low oxygen conditions. Using data from figure 1, support the researchers' claim. So having miRNA 466 promotes the transcription of GF.
Yes.
So the control cells are the ones that have the miRNA, correct? Okay. So we're going to say that the control cells have the miRNA 466 and they have higher levels of GF pre-mRNA than cells that lack miRNA 466, than cells that lack miRNA 466. Did they specify the oxygen conditions? They did say low, right?
Yeah.
Okay. D. Explain how the regulation of gene expression results in phenotypic differences among cells. So all your cells have the same genes, but those genes are going to be expressed to different levels. Some cells will express those genes, some will not. Some will express them higher, some will express them lower. Okay? So basically, different cells express different genes due to different transcription factors. So different genes lead to different phenotypes. In different cells.